RNA Transcription and Translation by Bhavya Mahesh and Disha Parekh. In order for DNA to be decoded and understood to build proteins, it must go through two processes, transcription and translation. In transcription, DNA gets decoded by an enzyme called RNA polymerase that builds an RNA strand based off of the DNA code. In translation, the RNA code gets decoded to make the actual chains of polypeptides in a ribosome. There are three parts to transcription. The first is called initiation. There is a section of DNA that is called the promoter. The promoter exists so the RNA polymerase knows where to first bind and start decoding. The TATA box is a common promoter sequence. It's a sequence of TA-TAAA attached to its counterpart, ATA-TAAA. The second step of RNA transcription is called elongation. So the RNA polymerase is a powerful enzyme that separates the DNA strands, reads the template strand, and creates the new RNA strand. The polymerase reads the nucleotides on the template strand and finds RNA nucleotides to pair with them. If there's a guanine on the template, there will be a cytosine on the RNA. However, if there's adenine on the template, there will be uracil on the RNA because thymine doesn't exist in RNA. The third step of transcription is known as termination. Just as there is a promoter so the polymerase knows where to start, there's also a terminator so the polymerase knows where to end. Once the polymerase reaches the section of DNA that is the terminator, the finished RNA strand, or the pre-mRNA strand, detaches from the DNA-RNA complex. However, before it can continue to the cytoplasm to code for protein, it needs one more step. Step 4, the last step, is called processing. We call this pre-mRNA because it's not fully stabilized yet. To make it stable, an enzyme adds a 5' cap containing guanine to one end and a poly A tail containing hundreds of adenines to the other end. The last step of processing is called splicing. Some sections of pre-mRNA are junk. They don't code for proteins. These sections are called introns. An enzyme called a spliceosome comes along and chops out the intron. The remaining sections are known as exons. These are the coding parts. The spliceosome connects them together, and finally, we have mature mRNA. This mRNA is the one that's ready to go out and code for proteins. Now, for part 2, translation. The mature mRNA strand drifts out into the cytoplasm, where it attaches onto a ribosome. So, in an mRNA, the instructions for building amino acids are the nucleotides, read in groups of 3. Each group of 3 is a code for one amino acid. The groups of 3 are called codons. There are 61 codons in total. Each of these are read to specify one of 20 amino acids. Just like there's a promoter in transcription, there's a start codon in translation. The codon AUG codes for the amino acid methionine and acts as a start codon to signal the start of protein construction. So that's great and all, but how is mRNA actually read to make a polypeptide? Transfer RNAs, or tRNAs, act as a sort of bridge that connects codons to the amino acids they code for. Each tRNA has a set of 3 nucleotides that bind to their counterpart codon. These are called anticodons. The other end of the tRNA has an attached amino acid specified by the codon. The ribosome is a structure made of protein and ribosomal RNA, also known as rRNA. It is where mRNA attaches and polypeptide chains are built. Each ribosome also has a subunit, the large top half and the small bottom half. The ribosome has 3 sites. These are called the A site, the P site, and the E site. So, what happens after the first tRNA is to attach to the mRNA? How does the chain grow? Well, we already know that the methionine attached to AUG on the P site. Now, another tRNA with an anticodon that matches the codon on the mRNA will come along and attach onto the A site. Then, the amino acid, which is in the P site, will attach onto the amino acid, which is in the A site. Lastly, the mRNA strand, which has tRNA attached to it, will move to the left. So, whatever was in the P site will now be in the E site, and whatever was in the A site will be in the P site, which means that the A site will now be empty. The empty tRNA in the E site will leave the ribosome and the process will begin again. This process is known as the elongation of amino acids. There's an easy way to remember the functions of all three sites, the A site, the P site, and the E site. Remember that the P site is where the polypeptide chain is forming. The A site is where the new amino acid attaches on, and the E site is where the empty tRNA exits. Finally, just as there's a terminator in transcription, there's a stop codon in translation. There are three different kinds of stop codons: UAG, UAA, or UGA. Once one of these codons enters the ribosome, it triggers the separation of the polypeptide chain from the tRNA, and the chain drifts out of the ribosome. This concludes the process of translation.